right, welcome everyone. Thanks to all for coming to today's Tools and Technology seminar. Hopefully there's ample pizza for everyone. I think there is, but I'll just do a little introductory stuff while lots of people are grabbing their pizza. I don't think we actually have any announcements today, but I do want to remind everyone there is an online audience. So if you have questions, please try to speak up so that they can hear your questions that much better. In addition, of course, if you're in the room, you got questions, just raise your hand like you typically would. If you're online and you have a question, please just put those in the Q&A pod, and we will be monitoring that and can pass those up to our speaker. So today, I'm pleased to present our speaker. We have Terra Sztain, who is Assistant Professor in Medicinal Chemistry and Biophysics. Thanks, Marcy, for inviting me and everyone for coming here.
I changed the title of my talk at the last minute, so hopefully no one came here specifically to hear about weight ensemble. If you did, I'm very happy to talk about it afterwards. But yeah, so I just joined. I started my lab last fall in the Department of Medicinal Chemistry and Biophysics as well. And so I wanted to talk about this preprint that we just posted a couple weeks ago, titled Machine Learning Driven Simulations of the SARS-CoV-2 Fitness Landscape. Hopefully this will be relevant to the audience here. So just a little intro. I'm very interested in proteins and how different sequences are controlling the dynamic structural ensembles that they fold into and how this is determining their function. And as you all probably know, proteins are really interesting, complex molecular machines. They're nature's catalysts. They perform a lot of interesting chemistries, and there's a lot of interest in designing them in order to create molecules of interest for human health and environment. Additionally, proteins are often the targets for a lot of different pharmaceuticals. So really interested in understanding these dynamic relationships so we can design enzymes and also inhibit them with different types of molecules. First I wanted to talk about the 2018 Chemistry Nobel Prize, which was Frances Arnold for directed evolution. And she was really inspired by the way that nature evolves enzymes over thousands of years. We have random mutations, and some of them end up getting lucky and providing better fitness, and these ones end up presenting in the population. And so what she wanted to do is say, can we speed this up? Can we be inspired by this process and do this in the laboratory in a controlled way? And so the way that this typically works is generating a very diverse library of many mutations and translating them into proteins, either in a cell-free way or inside of the cell, and then doing some sort of screening in order to select for fitness. And so this can be any type of process that you want, whether you want to optimize the catalytic efficiency, create some sort of new catalysis, but the important thing for doing this type of method is that you need some sort of screening or selection experiment so that you can trace back which mutants are leading to the protein that has the activity of interest. And then the way that this is carried out is iteratively you take the best performing mutant and then continue through several rounds of evolution. And so you're able to really traverse this vast sequence fitness landscape, which is very vast and difficult to grasp. And as you may know, I think, wasn't this yesterday that the Nobel Prize was announced for David Baker in chemistry? So this is the kind of a computational way to optimize the fitness of proteins. And so Rosetta is this software suite that has a lot of different functionalities for doing molecular modeling. And so a lot of them are based on using this, these physical descriptors of energy between atoms in order to determine fitness. So you start with some initial structure and you make some sort of perturbation, whether this is conformational sampling or mutation, and you evaluate the energy using this score function, and you can iterate this. And so you can do this for designing enzymes as well. And the way that this is measured instead of through activity is through, this score function. And the thought is that lower energy systems are more likely to have high fitness. So there's pros and cons to both of these methods. Obviously, one of the nice things about computationally doing this is obviously the resources are less and you can add sorts of constraints that you couldn't necessarily easily add in the experiment. One of the other caveats maybe of this just out of the box taking these generalizable score functions is this is an empirical descriptor that's been trained on the PDB. So when you look at the PDB, it's a lot of crystal structures and you don't have a lot of dynamic information. And when you have these sort of generalizable methods that are applicable to a lot of systems, you don't necessarily have the specific applicability. So they might need some sort of fine tuning in order to apply them to the system that you're interested in. So in my group, I'm really interested in how can we take information from physics and experiments, and how can we get the accuracy that you could get from defining things from physics first principles, but with the speed that you can get from doing machine learning from data, and how can we get the most information in order to apply these sorts of questions to questions that we're interested in for molecular design. And so what I wanted to talk about today was this interest in this project that started quite a while ago, but yeah, we just submitted the preprint onto bioRxiv. So SARS-CoV-2 is a really good example of protein evolution. So we know that when viral pathogens are evolving very quickly, we end up having a lot of data on mutations, and this can be really dangerous in the case of pharmaceuticals, where ending up evolution is evolving for resistance towards treatments and therapies and immune system, etc. So we wanted to ask this question, okay, can we take all this information we have from all of these sequences of SARS-CoV-2, and can we make some sort of predictive model that can predict future mutants of concern, so that we can start to prepare for them before they get out of hand. So one type of experiment that can be really useful that we ended up using for this is deep mutational scanning. So this, similar to directed evolution, involves generating a large library of many mutants, and in this case, and frequently they end up being expressed on the surface of yeast cells. So in this case, they expressed 100,000 different receptor binding domain mutants from the SARS-CoV-2 spike, and they wanted to look at how well these mutants are binding to ACE2. So they incubated these together, they tagged each of them with a fluorescent molecule so that then they could do this fluorescence cell sorting in order to get the relative binding affinity for all of these different mutants. So since we had 100,000 mutants, we went ahead and just created this really pretty basic machine learning model that can take in receptor binding domain sequence and output binding affinity, and just on its own, very simple, fully connected multi-layer perceptron performs really well. And just to confirm, we know that this relationship is not linear. You can't just take a linear model and be able to predict how these mutants are going to perform. And so this is really important because when you look at the sequence space that a protein can exist within, you have so 20 amino acids to the power of the length of the protein, so like 20 to the power 200. But if we're looking at just the point mutants, which some folks will do, it's only a couple thousand different variants, but you really can't make predictions or infer how individual mutants are going to affect the protein function when they're occurring together. So we looked at this and so our training set consisted of up to 9 mutants, mostly around 2 to 4 mutants. But we were able to, what we did was we looked at how if we just added or averaged the impact of point mutants. And when you do that, the error is very high, which is probably as you would expect, but the neural network continues to perform very well. So this is really useful for understanding the entire landscape, the sequence landscape that's possible for this type of protein, which we know is important because a lot of the variants of concern are coming out with multiple mutations, not just one. Okay, so when we looked at our model, we wanted to know if we could start to improve it. And we noticed that when you look at this region of high energy, we have a lot of scattered data points and we're not predicting very well. There's a lot of noise. And if you look at the distribution of the samples, you see that there's this really large peak. Sorry, this is the axes are before we transform the data from binding KD to delta delta G binding energy, but essentially we have these active mutants and then these inactive mutants, and they basically are just at the end of the experimental detection limits. And we wanted to incorporate this information, but we wanted to avoid training on the noise. So we thought that we really don't necessarily care about we don't necessarily care how the mutants are performing that are not folding or that don't have activity and just aren't binding. We don't really need a regression value for them. We just are interested in how well the binding mutants are. So we did this to see if we could improve the regression values of the sequences that actually do bind. And we either did a multitask training or we did a sequential training with a classification. And so we first classified them as to binders and non-binders, and then we performed the regression on those sequences, and we got slight improvement on those sorts of predictions. So that's something that we might be interested in cases, for example, like if we're doing these high throughput drug screening and we're only sometimes getting data on those molecules that actually bind. So there's a lot of cases where we think this could be useful. So next, we wanted to compare this and look at how doing just computing this with purely physics or with Rosetta. And so what we did was we modeled 1,000 mutations of the structure with Rosetta, and then we refined docking from this initial wild type structure, and we looked at how the docking scores correlate to the binding free energy, and it doesn't really correlate that well on its own. And so, however, there were 2 steps in this process. So the first step is the modeling of the mutants, and the second step is the docking. So we don't necessarily know whether it's that we don't have the correct mutants, or if just the docking score is not good enough to distinguish, the binders and non-binders and to correlate with the binding free energy. And because, for example, when we're modeling these in Rosetta, it's doing this sort of local refinement of where the mutation is. And we don't necessarily know because proteins, as mentioned before, maybe I can just, show this slide again, are very complex and dynamic. And so there are often cases where perturbations at one part of the protein are going to have impacts that are translated far away from the protein. And we don't necessarily know if that's going to happen. Also, of course, the context of protein folding isn't considered here because we know that the mutations are actually occurring before the protein is folded, so not afterwards. So this method of modeling isn't necessarily like 100% accurate. We also considered whether we should use AlphaFold. Also very exciting breakthrough showing how AI is and machine learning is relevant to a lot of biological problems. However, it's pretty well known, I think, at this point that distinguishing between variants within the same exact protein is not something that can be done very well. So when we did model some mutant structures with this a few years ago, it didn't perform very well, so we didn't move forward with that. However, there is literature precedence for showing that if you do represent proteins graphically, and in this case, we only had the structure of the wild type, we still had the context of, for example, if amino acids are far away in sequence space, but they're close in structural space. So we use the same edges, but different nodes to correspond to different identities of the amino acids to see if representing this graphically would improve our performance on predicting this. And it didn't necessarily improve. We're still working on seeing if adding structure and dynamics and other information can improve this. But because we had such a good, strong data set of many mutants and function values, and it trained really quickly with just a very basic network, we didn't necessarily need to add in the complexity of incorporating this structure. So we went ahead and said, okay, this performs really well. Can we use this model to make some sort of predictions? So what we did was we said, okay, can we assume that this sequence is following a Boltzmann distribution, and that say the probability of each sequence is related to the Boltzmann energy, which we're saying our fitness metric here is just the ACE2 binding, which we're getting, we're predicting from our model. And if we assume this, then can we run some Markov chain Monte Carlo simulations and simulate the fitness landscape of these protein sequences and get some information about this landscape. So we went ahead and ran these simulations. So we first proposed a new sequence with a random mutation at a random position, and we evaluated this sequence, either accept or reject with the Metropolis-Hastings criteria. So basically, if the proportion of the energy of the new proposed sequence versus the old proposed sequence is less than random normal distribution between 0 and 1, then we're going to accept it, and we're going to continue to run these simulations. And the first thing that happened is when you run this for many sequences, it just really quickly accumulated the maximum number of mutations. So this receptor binding domain portion is 200 amino acids, and we got close to that. We got saturating mutations, and that wasn't very interesting to us. And we also didn't have that that number of amino acids in our training set. In our training set, if you remember, this is the distribution of what type of mutations we would typically get from doing this type of experiment. So we wanted to essentially just make this a little bit closer to what we would expect, and we set a maximum number of mutations to 9 because that was what we got from the experiment. But then we saw the same thing happening where it really likes to quickly get to the maximum number of mutations. So we ended up adding this. We ended up focusing sampling near the wild type by increasing the proposal probability to proposing the wild type amino acid, and this is kind of analogous to the favor native residue bonus in Rosetta, where we don't want to go too far away. When we're looking at this sequence landscape, we don't want to just, traverse really far away. We want to really focus around the wild type, and there's so many ways to propose a sequence that's different than one that's similar. So we just increase the proposal probability. So now we have our simulations, and they're working pretty well. We optimized the temperature and the proposal, and we're getting something that looks reasonable to us, and we're able to look at the distribution of the sequence landscape of the receptor binding domain, and we find that when you plot this with UMAP, you can see that clusters are overlapping with the known variants of concern defined by the World Health Organization. So, and you can see that similar sequence variants are clustering together, and we even see that there is a cluster that overlaps with the sequences in Omicron, even though that wasn't trained on, and those mutations have many more than we allowed in our simulation, but they're still clustering similarly. So we thought this was exciting, that we can predict that we're aligning with what we see. So we started just with wild type, and we're able to simulate and predict that these mutations have high fitness and are likely to show up in the population. So here we can do just sort of sequence analysis on these, and what we see is that these mutants, which are very well known, like this E484K and the N501Y, are present in alpha, beta, gamma, mu, and E48K on its own is in iota and zeta. And we're also seeing several mutations that are present in Omicron showing up. So just looking at the wild type data, demutational scan, we're able to make these predictions. If you're looking at the sequence profile, you can see the next predicted sequence. So for example, this tyrosine is very common. This N501Y is showing up in all the variants, but the next most predicted is a phenylalanine. So this has similar chemical properties. So we think that the network is also able to learn these sorts of complex relationships that make biological sense. And then we can start to do, because we have these higher order mutations, we can look at how these mutations are co-occurring. We can look at the epistasis, which is really important for understanding this evolution. And in addition to predicting mutants that we have seen, we've also predicted several mutations that aren't seen in the population. Some of them have been found not in the variants of concern, but in other variants. And so we're hoping that this type of model can be useful for making predictions on which types of mutants should be focused on. So yeah, this is the conclusion. We have a lot of binding data from the steep mutational scan. We can train a very basic neural network on it. We can run Monte Carlo simulations, and we can make predictions about variants. We get a really good idea of the sequence landscape. Something that we are trying to incorporate to make this model even better, because this is really just data driven, is can we still incorporate some physics to make these predictions even better? Can we test these experimentally? Another caveat is that we're reducing fitness descriptions to just the ACE2 binding as the main driver of fitness, which there is some justification for in the literature that ACE2 binding, because it's the main function of the receptor binding domain, does control fitness, but we also want to consider this in the context of antibodies. So we want to know, are these mutants going to both escape antibodies and have higher ACE2 binding affinity? And we want to make sure that we're considering this not just from looking at how point mutations are going to affect this, and we really want to make sure that we're looking at this from an informed way, because a lot of the ways that they're doing this currently are just adding or averaging these impacts, which might not be useful. Because we have these simulations, we can evaluate temporal dynamics in the sequence evolution. We're really interested in incorporating more dynamic and physical features. And we want to apply this to other viruses and proteins. So we have a lot of protein engineering and other evolution of antibiotic resistance and antiviral resistance. We want to apply that and start building on that. So I wanted to share that here to see if anyone has interesting potential for collaboration, interesting systems, interesting data, as well as feedback. So I wanted to say this project started when I was a postdoc in Berlin with Frank Noe, but myself and Alex have been really pushing it forward in the past year to try and kind of build upon it and add some of these more complexities. And then there's a few folks in my lab currently who are also really interested in this. I have postdoc Sean, who's interested in seeing if we can apply this to other systems, and him, as well as Marissa and Yao, are interested in taking these sorts of ideas of high throughput experiments and synthesizing them with machine learning to make predictions in drug design with Michigan Drug Discovery. And then there's a few other folks in the group, like Daniel and Mortaza, who are doing a lot more of the MD simulations and looking at the physics and the dynamics, and how can we use this type of information to further understand proteins and understand which type of information is the minimal amount necessary in order to make predictions that are useful, but also in an efficient way. So those are all the slides that I prepared for this talk. I really wanted to bring this. This is my first time really presenting this project. So I wanted to bring it to this group and see if there's any discussion and questions or interest or feedback on this type of project.
Great talk. I was wondering, what does this landscape look like? Just like, how many of variants is could built, and each of size is made by the protons, and how about the landscape ruggedness? Is it rugged or is smoothly? And I know the epistasis is one of the evaluations. So how about how many of peaks of the whole landscape just look like? Yes.
Yeah, that's a great question. So, I don't know if I need to repeat that for folks on Zoom. You're interested in how the landscape looks like, how rugged is it, and how many peaks and valleys, and how does that relate to the epistasis? So one of the things, so we're able to look at this landscape. We're still thinking about what are the different ways that we can visualize this, because we really had to focus the landscape towards the wild type. And when you calculate this, so it's a 200 residue protein. So if you have 20 to the power 200, that's the entire landscape. So even within that, we can't sample the entire landscape. We're still sampling much more than can be done with the experiment, which is about 10 to the 5. And so we're again still focusing on, okay, so where can we sample, and we can extrapolate this information, so we can sample near what we know from the experiment, but we still can't get the entire landscape. And so we're looking at, and again, what we did find was that we should confine the landscape to between 0 and 9 mutations. So we basically have this sort of kind of view that's not going too far, but not going too close. And then we also can modulate the temperature parameter in order to determine how far away we're going from the different energy delta delta G values. And so we wanted to make sure that that was very close to the wild type as well, so that we're not falling too deep into a valley that we can't end up getting out of. And that's kind of another thing that we're looking at, because we're looking at this from like a thermodynamics perspective, using sequences. So there's a lot of stuff that we do in my group with molecular dynamics, and we're interested in enhanced sampling and how can we drive these simulations and start to figure out, okay, well, where is interesting to sample? We don't want to stay stuck in this, low energy well, but we don't want to just go randomly, and we want to, because we don't have an unlimited compute. So really great question. Yeah.
Can you talk a bit more about the training data you guys used for finding affinity prediction?
Yes. So the training data is from this deep mutational scanning experiment that was performed by Tyler Starr and Jesse Bloom. And what they did, so what questions do you have about the training data?
Like, I guess, what are the inputs?
Yeah. So essentially we have the sequence, and we have their binding affinity.
Okay, with like the ligand.
So yeah, with the protein. Yeah, so we have, very simple. We have sequences, and then we have values for binding affinity.
Can you say a little bit about how you controlled for overfitting to these data?
Good question. Great question about how to control for overfitting. And so I think one of the things that's really difficult to tell, especially when you're doing these high throughput experiments, is how this question about, are you just making predictions about this? And we actually did look at other deep mutational scanning data sets, because Jesse Bloom and Tyler Starr did perform these starting from, and I didn't show the slides for that here, unfortunately, but they did start once Omicron came out, once, beta and delta, before Omicron came out, they did actually redo these deep mutational scanning experiments to start to look at the how is the sequence landscape when we're starting from this other sequence. And so one of the things we noticed really quickly when we tried to just like aggregate this, I wish I could, oh, this isn't my computer, I can't show you other stuff. So one of the things we noticed is we definitely had to find some way to normalize between the experiments, because there's just a lot of noise and a lot of error. And we also noticed that when they started to perform the subsequent experiments, and this is why I wanted to emphasize the importance of the higher order effects, is that they wanted to more quickly predict the impact of point mutations. So instead of just doing this random mutagenesis and generating 100,000 mutants, they actually did like 10,000 mutants, so they could just get the point mutants. So we had a lot less data to train on. But I'm not really answering your question about overfitting. And I think one of the things that we wanted to consider was splitting the training and test and validation data more thoughtfully. Right now it was just a random split, and we ended up happening to get lucky that we didn't actually train on the variants of concern. We didn't even train on single point mutants of these mutants that are really interesting. We were able to predict them with the model, even though they weren't seen in the training set, but that was just by happenstance. So we're interested in seeing if there's more creative ways that we can determine if we're extrapolating, you know, better on unseen data rather than overfitting. That answer your question?
Maybe another question, in publishing these potential variants of concern, obviously they can be super useful for rapid response if they're seen in the wild. But on the other hand, these are potentially more virulent sequences. What sort of precautions did you consider in terms of disclosing these to the public?
Yeah, that's also such an interesting question, because so one of the things about doing this type of experiment in this way that's not like, for example, a gain of function study where people are just like actually evolving the virus in order to be more powerful, which can obviously be dangerous. And we, we're doing this in the computer, which is great. But even if we do this, in vitro, and we're just expressing, these little pieces on their own, this isn't necessarily something that's going to be released. But yeah, if we make these predictions and we say these are likely to be more virulent, I guess there is a chance that someone can use this maliciously. I think that's maybe a topic in conversation, but I think because of the limit to which these are, where these are just within the spike, and we know that the mRNA vaccines can easily be deployed with, and we can just easily design different mutations in the spike. I don't think they're likely to cause issues, but if you have any suggestions regarding that or other comments, I'd be happy to hear them.